Hi, hello, good evening everyone. I am Sathish. I am biology teacher. I had 10 years experience. I am handling 11th and 12th standard. So today's session we are going to discuss what is rDNA technology. What is rDNA? We know very well what is DNA. DNA is a deoxyribonucleic acid. Is a new one. What is rDNA? Yes, it is recombinant DNA. Recombinant DNA. What is recombinant? Is a altering of genetic material from one organism to another organism. If fusion of two different DNAs are called as rDNA technology. If we are select one DNA from the donor cell. Okay, this is a bacterial. The bacterial cell is called E. coli. E. coli means is Escherichia coli. Is Escherichia coli having normal their own genetic chromosome material. Is addition with another one more chromosome is called plasmid. This is plasmid. What is plasmid? Plasmid is a circular double-stranded self-replicating own DNA is called as plasmid. Here plasmid is used as a vector. What is vector? Vector is nothing but a carrier of a genetic information from one organism to another organism. One more organism we are selected from, which may be human cell, plant cell or even animal cells. Okay, is a selective or desired or suitable replicate DNA from a host cell. Okay, this is a recipient cell and this one is the host cell, which may be any kind of cells. Okay, desired DNA. Okay, so isolation of desired DNA from the host cell. Okay, this is a desired DNA and this is a plasmid. Isolation of plasmid from Escherichia coli. Okay, using restriction endonuclease. RE means restriction endonuclease to cut the DNA, which is also called as molecular scissor. So to cut the DNA into two separate strands, same as this DNA also is a cutting of several pieces. Okay, after cut the DNA, which is fuse the two cutted pieces. No, the two cutted pieces are joined together. So the isolated DNAs are fused together. So two different genetic material are fused together. No, that is called as rDNA. Why we are fused two different DNAs? Because this is from bacterial cell. What is the nature of bacterial cell? Which is fast replicating cell. Especially the Escherichia coli is every 40 minutes once replicate their own genetic material as well as cells. A single cell is divided into two. You know very well, bacterial cell is undergoes binary fission. It is divided into two cells. It is a fast replicating cell. Why we are selected the host? The host may be, for example, nowadays the sugar patients are more. Okay, the 70% of members are undergoes diabetes. So how to cure that? So in nature, our body, the pancreatic cells produce naturally insulin. What is the role of insulin? The insulin to control the blood glucose level. Okay, but the enormous amount of sugar patients, it's not sufficient naturally produce the insulin. So we are choose the rDNA technology to provide more and more amount of liters of insulin injections every day. Okay, so through only the rDNA technology. How it is? For example, this is a pancreatic cell of a human being. What is the role of pancreatic cell? The pancreatic cell, the pancreatic juice which secrete the insulin. We know very well what is insulin. It is a hormone to control the blood glucose level. So we are select one DNA, no, this DNA to produce the insulin. So insulin produced DNA are isolate from a host cell, and the fast replicating plasmid DNA isolated from the recipient bacteria. Both are fused together to form a rDNA. Okay, we are fused the rDNA. How it is multiply or produced? Again, the same Escherichia coli bacteria, the rDNA which introduced into the same Escherichia coli bacteria. So we are isolated the different DNAs from, one is from recipient DNA, another from donor DNA. Both are fused using DNA ligase. We know very well the DNA ligase to fuse the DNA fragments. The fused DNA fragment is rDNA. The rDNA introduced into E. coli bacterium. What is the role of E. coli bacterium? Every 40 minutes once it is replicating into 2. Okay, so in a 1 day or 1 or 2 days it is fast replicating and produce thousands and thousands of bacterium. What is the role of the DNA, host DNA? It is produce insulin. So as well as the bacterial DNA is multiplying and the production of more amount of insulin. So every day life we are using the insulin hormone produced through this technology only. So this is a very important technology for nowadays the biotechnological field, especially the recombinant DNA technology. I hope children you are like this video. If you have any doubt, please put in the comment box, share this video and please subscribe our channel. Thank you.